So we are going to continue on in this folder and talking about the immune system. With our last folder, we looked at the innate system and some of the components of it. And you probably remember that this is the system that is in place from birth that allows you from the time of birth to defend your body against microorganisms. I mentioned when we first got into the immune system that there is a second branch, which is known as the adaptive branch. And that's where we're going to go today and spend the rest of our time in the immune system. So a little bit about this branch. One of the things that I said about it when we first started into the immune system is that it is not fully functional from the time of birth, but it is adaptive, meaning that once you're born, it begins to adapt to recognizing the microorganisms that are out there, that are in your environment. One of the things that's also really unique about this particular branch is it is specific. So what I mean by that is it is able to tell a virus from a bacterium. It doesn't see things as just foreign and harmful, but I don't necessarily know what it is. It recognizes them. It's also able, for example, to tell one virus from another. So for example, it could tell human papillomavirus, HPV, from human immunodeficiency virus, HIV, from varicella virus, which causes chickenpox. And in addition to being able to recognize those different viruses, it has different cells that are parts of this particular system that are going to fight each one of those individual viruses. It's also capable of telling bacteria from each other. So it would be able to tell the difference between a staphylococcus and a streptococcus and a tuberculosis bacterium. And once again, it's going to have specific cells that are actually adapted to go after each of those bacteria. So again, today we're just kind of getting an introduction to the adaptive system so that we're really prepared next week to kind of get into this system more and some of the specifics and the cells that are involved in it. One of the things that you should know at this point, though, about this particular system, the adaptive system, is it has a couple of branches to it. So the first of these branches is what's known as the humoral branch. And humors is an old term, kind of an old-fashioned term for body fluids, meaning things like lymph, things like blood. So the humoral branch is the branch of the adaptive system that's functioning in body fluids. It's functioning in the blood, it's functioning in the lymph. We talked, when we first got into the innate system, about the fact that some viruses can actually get into cells of the body and take over the cells and cause them basically to become a virus factory. Once a virus or a bacterium or another microorganism is actually inside another cell, it's capable of hiding from the humoral branch. So the humoral branch is only going after things that are floating around out in the open, if you will, in the blood and in the lymph. But because, as we've already established, some things are able to get inside of cells, we also need a cell-mediated branch of this system to go after those things that are hiding out in a cell. And that's where this cell-mediated branch comes in. So this is really kind of a basic introduction to the adaptive system, what it does, the fact that it has a couple of different branches. What I want to do now is continue on with just some basic introductory information to help you understand how this particular system is kind of working in general before we start getting into the specifics of the different branches. So with this particular slide, I want to talk a little bit about self-antigens, and it's probably important before we do to establish why self-antigens are actually needed. I mentioned with the adaptive system that it's capable of telling what things are foreign to your body, and it's actually capable of differentiating between all of those foreign things. But if we're going to be able to tell that something is a tuberculosis bacterium, for example, we need to be able to know or be sure that we know what tuberculosis bacterium looks like compared to what the cells of your own body looks like. So one of the things that we have functioning in the adaptive system are what are known as self-antigens. And they are going to help the adaptive system recognize what's you from what's something else that may be in the body, a streptococcus bacterium, some other cell that actually is foreign and shouldn't be there. So here's a little bit about self-antigens. They are just small proteins that are on the outside of your cells that denote your cells as a part of you. So all of us have these little proteins. They're of slightly different shapes because none of us are genetically identical, unless you have an identical twin, and then they would have these exact same self-antigens with the same shape as you do. These self-antigens are not foreign, they're not antigenic to you, but they would be highly antigenic to anyone else. So you may be familiar with organ transplants. And one of the things that we always have to worry about with an organ transplant is that that organ is going to be rejected. What that means basically is that the host, so the person who received that organ, their body, and specifically their immune system, recognizes the self-antigens on the donated organ. Because if it's a donated heart that comes from me to you, that heart is going to have my self-antigens on all of its cells. And if your immune system isn't suppressed through medication, what's likely going to happen is at some point, it's going to recognize those self-antigens as not a part of you. So your self-antigens denote you as you. They are specific to you, so yours look like nobody else's on earth unless you have an identical twin. But those same self-antigens are highly antigenic to anyone else. Somebody else's immune system would recognize them as not a part of them, as something that's foreign. And because it's foreign, it's something that's potentially harmful. So one of the things that I want you to know about these self-antigens is that they are displayed on what are known as MHC proteins. So that stands for major histocompatibility proteins. If you look at this picture over here, I've tried to draw kind of a simplified drawing of maybe some cells that are in my body. And I'm using this picture of me, okay, to denote my self-antigens. And right here, we've got that MHC protein. So the self-antigen is displayed, it's attached to this MHC protein. I very often think of the self-antigens as a flag and the MHC protein as a flagpole. And basically what we've got going on in our bodies at all times is we have immune system cells that are crawling over all of our cells, and they're looking for these MHC proteins and what the MHC proteins are actually displaying. And as long as they see our self-antigens, that lets them know, hey, this cell is good, it's okay, it's a part of our body. When they come to a cell that is displaying something different on its MHC protein, so what you would see here, for example, that doesn't look like the rest of the self-antigens, that's going to let the immune system know that that's a cell that is foreign, that's not a part of us. Because of that, it could potentially be harmful, and it's something that likely should be destroyed. So with the last slide, we started talking a little bit about MHC proteins, and now I want to give you some additional information about these MHC proteins. So there are actually two types of MHC proteins that we find on the cells of the body. The first of these is what's known as a class 1 MHC protein, and these are found on all cells of the body. The only exception to this would be red blood cells. So they're found on muscle cells, they're found on nerve cells, they're found on the cells of your organs, on skin cells. Basically every type of cell in the body has these, with the exception of red blood cells. Here's my representation of a cell in my body that has a class 1 MHC. So this is a healthy cell. This could be my liver cell or my bone cell or an organ cell or whatever. But here's that class 1 MHC. And as long as this is a cell that's healthy, and what I mean by that is it's not taken over by a virus, it's not harboring something that could potentially be harmful to the body. As long as this is a healthy cell on its class 1 MHC, it's going to display self-antigens. Now remember that viruses and some bacteria can actually get into the cells of our body and can actually take those cells over. And when they do so, those viruses or whatever it happens to be is going to inject some of its DNA into the host cell DNA, And what that means is if this is a cell in my body, we could even say it's this cell, and it has become infected, now we've got viral DNA in my cell. And that viral DNA is going to change the shape of the self-antigen that's displayed on the MHC class 1. And all of a sudden, that flag that's displayed, if you will, or the self-antigen that's displayed on the class 1 MHC is going to look very different than a self-antigen. So that when an immune system cell crawls over it, it's going to see, hey, this is not self. This looks different from what should be displayed on class 1 MHC in a healthy cell. And that's going to let the immune system know this is a cell that has been taken over. This is a cell that's not healthy. This is a cell that we need to destroy because it's going to potentially be harmful to the body. We also have a second type of MHC protein that's found on some cells of the body. So these are what are known as class 2 MHC proteins, and they are found only on very specific cells that are a part of the immune system, which are known as antigen-presenting cells, or APCs. APCs effectively are the policemen of the body, if you will. So they're traveling throughout the body, they're traveling through your lymph, traveling through your blood, traveling through your tissues, looking for pathogens, things that are potentially foreign and therefore harmful to the body. And when they find something that they believe to be foreign and harmful, what they're going to do is they're going to engulf that thing through phagocytosis. They're going to digest it, and then they're going to spit up some of the proteins from that foreign harmful substance onto their class 2 MHC proteins. So if you look at this picture down here, this is a representation of an antigen-presenting cell, or an APC. Like all other cells of the body, antigen-presenting cells have the class 1 MHC that is going to display the self-antigens. However, when an APC has encountered something in the body that it determines to be harmful and foreign, it's going to phagocytize that substance and then spit out, proteins from that substance and display them on the class 2 MHC. The reason for this is when these APCs find something that is harmful, they are going to approach another immune system cell, which is known as a helper T cell. And they are going to show the helper T cell this substance that they found in the body that's harmful. Helper T cells are kind of like the generals. So they're going to see this potentially harmful thing that's displayed on the class 2 MHC proteins of the APC, and they're going to make a decision whether we activate the entire immune system against it or whether it's something that's actually harmless. But if you think about an APC that has phagocytized a bacterium or a virus or something else that it found in the body that's harmful, and it's now displaying proteins from that harmful substance on its class 2 MHC, it's very important that we also have the class 1 displaying self-antigen, because when that helper T cell sees this APC, when this APC displays its antigen, its harmful substance, to the helper T, we want the helper T to first see the self-antigen on the class 1 MHC so it can say, okay, same team, this is a good guy. I don't want to destroy the APC because I know it's a part of my body, but it's also showing me that there's something that's harmful and foreign in the body that looks like this. If you have some type of microorganism invader in the body, there's not just one, there's hundreds, thousands, potentially even millions. And so we want to let the immune system know this is what it looks like. You need to be on the lookout for it. If you see it, destroy it. And alerting helper T cells to the presence of this substance is the first step that we have in kind of coordinating and activating the entire immune system against harmful microorganisms. But again, really important when an APC is doing that alerting, that it has the class 1 MHC, that it has a self-antigen that it can display so that the helper T doesn't think the APC itself is actually a cell of the body that has been taken over by a microorganism. So as we get more into the adaptive system, you're going to start hearing a lot about B cells and T cells. B cells are also known as B lymphocytes, and T cells are also known as T lymphocytes. So a lymphocyte is a type of cell that you've learned about before. It's actually a type of blood cell. And so because these are blood cells, like all other blood cells, they actually originate in the bone marrow. However, T cells get their name because once they've kind of got to a certain place in maturation, they're going to migrate from the bone marrow to the thymus, which you may remember is that lymphoid organ that kind of overlays the heart, and they're going to complete their maturation there. And that's actually why T lymphocytes or T cells are known as T cells, because it stands for where they're actually developing, which is in the thymus. The other major type of cell that you're going to start hearing a lot about is, again, what's known as a B lymphocyte or a B cell. B lymphocytes or B cells complete their development in the bone marrow, and that's the reason for their name, B cell or B lymphocyte. A lymphocyte, whether it's a B cell or a T cell, while it's developing, while it's maturing in either the thymus or the bone marrow, has to become capable of doing a couple of different things. So the first thing that we need a lymphocyte that's maturing to do is to become what's known as immunocompetent. Immunocompetent means that that cell is able to recognize its one specific antigen by binding to it. And there's a very unique thing that happens in the thymus for T cells, in the bone marrow for B cells, that allows these cells to be immunocompetent. That is, these cells that are developing sprout on their surfaces a receptor that's kind of a random shape. So if you look at lymphocyte A here, my representation of a lymphocyte that's developing in the thymus, you'll notice it has sprouted a receptor that has kind of a V-shaped end on it here. Here's lymphocyte B, just my example of another lymphocyte that's developing. When it becomes immunocompetent, I'm showing it as developing kind of this square-shaped receptor on its end. And then here's another one, lymphocyte C, which I'm saying is specific for tuberculosis bacterium. And when it becomes immunocompetent, it's going to sprout a receptor of a different shape. You may notice that this first lymphocyte with kind of this V-shaped receptor end is immunocompetent for flu. And the reason that I say that is because its receptor is of a shape that's going to allow it to bind to virus. It's going to recognize flu virus. Now, when we talk about B cells or T cells and we say that they're recognizing something, they don't do this like you or I would recognize something, which is by seeing it and remembering it. So these cells don't have eyes. When we say that a cell is capable of recognizing an antigen, what that means is it has a receptor that allows it to bind to that antigen like a lock and key. So lymphocyte A here is immunocompetent for flu virus because it has a receptor of a shape that allows it to bind to the flu virus like a lock and key. Over here, we have my example of another lymphocyte. This one is lymphocyte B, and I've said that it's immunocompetent for HPV. And what that means is it has a receptor that's of the correct shape and size to be able to bind to human papillomavirus. This one over here, again, is immunocompetent for tuberculosis bacterium. Here's my little representation of tuberculosis bacterium. Of course, it isn't a blue circle, but it has a specific shape, and the receptor that is sprouted on the surface of a lymphocyte that's immunocompetent for tuberculosis bacterium is going to have a shape that allows it to bind tuberculosis bacterium like a lock and key. So once the cell has this receptor of a certain shape that allows it to bind a specific antigen, we consider it to be immunocompetent. I always get the question, so these cells are developing in the thymus, they're developing in the bone marrow, maybe before you ever see flu virus, or maybe before you ever see HPV. Maybe you'll never get exposed to HPV, but your body right now has lymphocytes that are specific for all the different kinds of HPV in it. The way that this happens is we have hundreds of thousands, even millions of different lymphocytes that are developing in the bone marrow and the thymus. The receptor shape that they sprout is just kind of random, but there's so many different random shapes that we have one for everything that could potentially be out there. If we did not have a receptor for a certain substance, that would mean that we would be unable to hit. We wouldn't have any cells that are immunocompetent to it, and that means that we can't have any type of a reaction to it. That's why it's always concerning anytime there's a swine flu or a bird flu, because what that means is this is a strain of the flu that developed in another species and jumped to humans. And if it developed in another species, we have no idea what its shape might be, and that increases the chances that we're not going to have a receptor that's going to be able to bind it. So that's why there's always a panic when we've got swine flu or we've got a new avian flu, because we're just not sure how well the human body is going to be able to react to it. The other thing that a T or B cell has to do as it's developing in the thymus or the bone marrow is we have to ensure it's self-tolerant. So with the immunocompetence, remember, it sprouted a receptor of a specific shape that allows it to be able to bind to something like a lock and key. We want to make sure through this second process, through this check on self-tolerance, that what that cell's receptor is able to bind to, like a lock and key, is not your own self-antigens. Because if it is your own self-antigens, it's going to destroy cells that are displaying your self-antigens. So what we want to do in this second process as a T cell or a B cell is maturing and it's becoming a fully functional immune system cell that we can send out into the body to defend the body against microorganisms, is look at this self-tolerance. And there's a two-step process that ensures self-tolerance. So the first step in the process is what's known as positive selection. And I want to walk you through a little bit what's going on with positive selection. So we have a cell here. This would be a cell that's in the thymus. Specifically, it's an antigen presenting cell. What you're seeing right here is a class 1 MHC protein, and then sitting right in the middle of that class 1 MHC protein is a self-antigen. What we've got over here, this cell in kind of the turquoise color, is a cell, a T cell that's developing, that's working on becoming mature before it gets released into the body. And what I want you to know is this antigen presenting cell here is showing this developing T cell, the self-antigen on the class 1 MHC. And you'll notice this T cell has a receptor that does not allow it to recognize the class 1 MHC. So again, when we talk about cells being able to recognize something, they can't see it, right? Because they don't have eyes. But recognizing means that we need these structures to bind together like a lock and key. And you can see that's definitely not happening here. If that's the case, this cell is a problem, because if it cannot recognize, if it can't bind to class 1 MHC, that means it has no idea which of all the receptors, which of all the proteins on the surface of a cell of the body is displaying your self-antigen. So it cannot recognize self from non-self, and that's a problem. So in this first step of positive selection, if the cell isn't able to recognize the class 1 MHC, okay, where again, self-antigens are displayed, this cell is going to go through apoptosis, which basically means this cell is going to die. We can't release it into the body. It's too dangerous because it cannot determine where self-antigens are displayed. What we want to happen in positive selection is what you see in this picture here. So again, we've got an antigen presenting cell. It's got a self-antigen being displayed on the class 1 MHC. Here's the cell that's developing in the thymus. It has become immunocompetent. Now we want to make sure it's self-tolerant. And you'll notice it's recognizing that class 1 MHC in that it's binding to it perfectly like a lock and key. This is a cell that we want so far. We're going to send this cell on into the negative selection because it can recognize class 1 MHC, which means it knows where to look to see self-antigens. So the next step in the process for cells that are capable of recognizing the MHC proteins is what's known as negative selection. What we don't want to happen in negative selection is what you see in this first picture here. So here is our APC. It's got the class 1 protein, MHC. Here's a little self-antigen. This is our developing immune system cell. It's able to recognize the class 1 MHC, and it's binding to it like a lock and key, but we've got a problem because it's also binding to the self-antigen like a lock and key. This is not something that we want, because what happens is this cell, if it goes out into the body because it recognizes self-antigen and binds to them tightly, is going to start working to destroy cells that are displaying self-antigens. So this is not what we want to happen in negative selection, and any cell that does this is going to go through apoptosis. It's going to die because it is not going to be a good immune system cell that's going to not attack and not destroy your own body cells. If you look at this picture down here, so this fourth picture, this is what we want to happen in negative selection. We've got our APC, it's got the class 1 MHC that it's displaying. Our developing cell recognizes the class 1 MHC. Again, that means it's binding to it like a lock and key. However, it does not bind like a lock and key to that self-antigen. So it's not going to activate against cells that are displaying self-antigens. So again, basically what we want to do through this self-tolerance process is we want to make sure that our lymphocyte that's developing knows where to look for self-antigens, so it can recognize one MHC, but we want to make sure it doesn't bind tightly and react against self-antigens. So it can recognize, know where they are, but it doesn't react against them. Only about 2% of cells make it through positive and negative selection. 98% either can't recognize class 1 MHC, so they have no idea where to look for self, or they recognize the class 1 MHC, but they also recognize the self-antigen, and they would react against cells that are displaying that self-antigen. So 2% of the cells actually make it out into the body to become functioning immune system cells.